Thank you. And thank you again to the organizers, Michelle and Ryan, for helping me get here. This is my first time in Africa, and certainly South Africa, so it's been a pleasure and a delight to be here. For those of you who know me, you know that I'm a recent transplant to the Netherlands. My former institutions and where I have current affiliations are the Woods Hole Marine Biological Laboratory and also Brown University. So I am a microbial ecologist. Oops, I'm doing this wrong, sorry. I'm a microbial ecologist by training, and I consider myself very lucky because among the fathers of biodiversity, E.O. Wilson, I think I'm envied in that one of his quotes from an autobiography, he stated, "If I could do it all over again and relive my vision of the 21st century, I would be a microbial ecologist." So I certainly agree with that, and in fact on the book that's shown here, there is in fact many microbes represented. This is just a drop of water that's on the cover, and if we think about a drop of water in terms of 1 mil, there are many, many microbes that are present even in such a small little microenvironment, to the tune of about 1 million bacteria and archaea, 10 million viruses, and 10,000 protists. And if we compare that to the estimated number of stars in the universe, in just the ocean, we see that there are many orders of magnitude more microbial cells in the ocean than in the stars in the universe. So today what I'm going to do, although my research has spanned all 3 domains of microbial life, I'm going to focus today on the protists, sometimes called kingdom Protista. And they offer a great diversity of morphology, which you can see here. So there's a very rich level of morphological diversity that they represent. But also, protists range in size and lifestyles and trophic levels. Now, the first to sort of define the Protista was Ernst Haeckel himself, and this is the famous 3 kingdoms of life, wherein Protista were sort of what was left over between the plants and animal kingdoms. And but he was sort of famous for representing it in the form of this tree. But the concept of protist has really evolved over time, and folks may have some of us in the room at least have grown up with the 5 kingdom concept of protists, where you have plants, fungi, animals, protists, and Monera. Of course, this was superseded. This lasted for a couple decades, but was superseded, of course, by the work of Carl Woese and Sogan and others into the 3 domain sort of concept of life. And this is shown here, wherein protists really became realized as a very diverse set of eukaryotes in the tree of life. And this is a picture that is now established, and even in textbooks, we understand it to sort of be the natural organization based on phylogenetic relationships of organisms that are present in the environment. And among the Protista, the higher order relationships are still somewhat in flux, but are also starting to stabilize. And so things that were once considered maybe families or orders are actually now given phylum level in many cases. So the realization of the diversity that's present in the Protistan world continues to be realized. So I grew up from a molecular systematist looking at obscure protists that live in the ocean, understanding the importance of the ribosomal RNA molecule as a metric for looking at microbial diversity in microbes, partly because many of them don't have a morphology to differentiate them, but also because a lot of the organisms that we work with can't be cultured. So when we think about the diversity of life, it's important to understand that multicellular life takes up a very small portion of the total diversity that's present on life. And in terms of Protista, my postdoctoral advisor and somewhat graduate advisor, Mitch Sogan, was really responsible for filling in a lot of the diversity that is understood now among the Protista. And of course, his mentor, Carl Woese, was responsible for the discovery of archaea. And Norm Pace up here, I've placed on this tree because he was really one of the first scientists in his group to start thinking about environmental sequencing using this now dated approach of phylogenetic diversity studies wherein we amplified entire genes and cloned them and sequenced them and inferred unknown phylogenetic placement of organisms in the tree of life. I was able to sort of do this as a postdoc, an early postdoc in Mitch Sogin's lab, and this was one of the first attempts at trying to quantify, not so much quantify the diversity, phylogenetic diversity in an extreme environment. This happened to be a very acidic river in Spain where I first worked as a postdoc, and we found that despite the extreme conditions, there was a variety of different protists and other eukaryotes that lived in this river. And so this really opened up the floodgates of being able to use these environmental DNA approaches to look at the diversity of protists that are out there. But here we see the tree of life that was published in 2015. And it really is very modestly represented by microbial life. And of course, we're still challenged as microbial ecologists with the fact that we have so few named species. And in the bacteria and archaea alone, there's about 10,000 named species only. And within protists, that number is sort of fluctuating a bit, but anywhere between 100 to 200,000 named species, I think, is somewhat accepted. But we understand that there's a lot of cryptic species that have been unrecognized. So in an attempt to start to uncover this hidden diversity of the hidden majority that's out there in the environment, I was part of an effort back in 2004 that was spearheaded by the Census of Marine Life. That was a 10-year effort to look at the number of species that live in the ocean. And so as part of this effort that was led by Mitch Sogin at the Marine Biological Laboratory and Jan de Leeuw at my new institute, the Royal Netherlands Institute for Sea Research, there was an effort, a community effort to start to sample the diversity of microbes that live in the ocean. And I'm not going to talk at all about these results. Suffice it to say that we didn't do such a good job in that we estimated that we only sampled about 1 divided by 10 to the 18th the volume of the oceans. So this continues to be a challenge, is not only being able to sample deeply, but sample broadly geographically to understand the diversity of microbes that are part of the environment. Now, one of the legacies of ICOMM, though, as it was called, the International Census of Marine Microbes, was that we really did establish a method, I think, that has really taken hold of many different communities that began in the marine realm, wherein we use this tag sequencing approach. So instead of sequencing the entire gene, we only targeted a variable region. And at the time, constraints of the technology, the 454 technology that we used, required us to target a small molecule, a small part of the molecule, I should say. But of course, this methodology, as everyone is aware and increasingly aware, relies heavily on a reference database. And of course, ribosomal RNA has served as that database to many and continues to do so. But as we recognize, it's not a phylogenetic tool. So we don't use it to define new taxa per se or to place them phylogenetically in a tree, but instead we recognize that they're novel and use other methods to get more refined phylogenetic placement of those taxa. So simultaneous to the census that ended in 2010, I had a separate project that was called Microbial Inventory Research Across Diverse Aquatic LTERs, also known as MIRADA. And this was a project that was across 13 of the aquatic LTER sites. And it was a very much inventory research, but at the microbial level. And we aimed to document, describe, and discover diversity across these diversity of habitats that are known, of course, to house many different microbes as well. But one of the things that MIRADA did was, in addition to looking at bacteria, my group also helped refine the bacterial primers and establish primers for archaea and also eukaryotes. And again, at this time, it was a time when we were restricted by the length of the molecule we could target. So for eukaryotes, we chose the V9 region, and we did that to aim to try to capture protists. But these V9 primers will actually capture all different types of eukaryotes with better or worse resolution and differentiating them with those limitations. So of course, this methodology was adopted by other groups who have done much more extensive sampling of the oceans, such as the Tara Oceans Survey. And the exciting thing about this work is that it's really probably the largest survey so far of particularly protist planktonic species, because that was more of a target of this group. And the estimated number of protist planktonic species that this group came up with was 127,500, again, based on estimated diversity measures. And, but back in 2012, it was established that there should be a common barcode for protist species overall. And a group came together that was led by Jan Pawlowski, who is joining us today in the conference and this week. And it reviewed all the different barcodes that were used for specific groups of protists, but in the end recommended that a certain pre-barcode be used, and that was the V4 region of the ribosomal RNA gene. Now, what they also did was establish what the recognized numbers of species were, which was a big range for the different groups, and also estimated between 140,000 to 1.6 million different species of protists as part of this effort. So this is the sort of schema that was recommended, was either picking individual cells or, when possible, of course, culturing them, and using other means for those that were uncultivable, and then using this sort of first stage pre-barcode followed by species-specific barcodes that are available and being developed for different groups of protists. But as you can imagine, this is sort of necessary, right? Because it is really a, the phylogeny of protists is so diverse that it really does require a diversity of different barcodes that are present for different groups. So one of the really exciting developments that's happening in this community and in my community is that the taxonomy, the realization of all this environmental survey work has left with many of us at the real need for having an established and unified taxonomy. And this is something that the community has been working at for a very long time, but not very effectively. And at last, there has been agreement that these groups of experts would come together and establish a unified taxonomy. The effort is called UniUke, and it's an effort to really achieve a universal framework. And it's funded through various sources, including the Moore Foundation and the International Society of Protistology. And this is a recent announcement of this effort. I think it's really timely and somewhat overdue in some cases, but nevertheless, it's a community effort, and part of this effort involves establishing a unified phylogenetic framework, a standardized taxonomy, whether or not it's going to be stable, but at least agreed upon and used exclusively by people that are working on environmental data sets. But furthermore, there's this development of this thing called the Uke Bank, which is an effort to harvest all the V4 18S ribosomal RNA gene data sets in order to establish what is really novel out there among these various types of environmental surveys that are being conducted. And there's a call, an open call for anyone that has these data sets to contribute to this effort, and I really encourage any of you yourselves, if you're doing this kind of work, or sharing this with your colleagues to contribute to this effort that is really quite important. Okay, so I would like to spend the rest of my time giving you a couple of examples of maybe pitfalls in the process of trying to establish barcodes within the kingdom Protista, within protists. Now, I mentioned that protists range dramatically in size. They actually can go 4 orders of magnitude, but the examples I'm going to be giving you span 3 orders of magnitude, from very small algal cells called haptophytes to the very large macroalga called sargassum. And in my new position at NIAS, part of what I'm going to be focusing on, not exclusively, but in part more intensively, are photosynthetic microbes such as these. So first I want to mention the haptophytes. They are a very diverse group. They're actually a phylum in and of themselves. And the group that I've been focusing on is the order Isochrysidales. And some of you may not know of them, but know of them from examples of these white calcareous blooms that develop off of off of England, where they actually can be visualized by space. This is produced by this calcium carbonate containing Emiliana huxleyi alga. If any of you have visited the White Cliffs of Dover, this is one of the organisms that's partly responsible for those, for that entity. But for a long time, these haptophytes were thought only to dominate in marine environments, but it turns out there are also freshwater lakes in which they are very much welcome and abundant and even bloom at times. In fact, we discovered an entirely new lineage of haptophytes that are found in high latitude lakes such as Greenland. Now, in addition to being interested in their phylogenetic diversity, one of the really unique applications of these organisms is that they possess these special lipids that act as paleothermometers, because the number of saturated bonds in the lipid molecules that they produce will actually effectively record the temperature. So they're correlated with temperature. This has been known for about 25 years and has established a part of paleoclimatology that is widely used now. And there have been different proxies used for marine and freshwater environments. But one of the challenges with freshwater environments is really being able to understand the diversity present in the environment to be able to make those correlations with the proxies. And so in collaboration with my colleague, Youngsung Hwang, who's a professor at Brown, and various lineages of students that have helped with this project, we've been addressing the questions that I show here, such as what is in fact the global diversity of these haptophytes that produce these unique lipids, how is it recorded in lake sediments, and finally this one group that we find is hyper-diverse. We're asking the question, why is this one group so diverse? And the reason that's interesting, I'll get into a minute, but the methods that we use range from not only looking at DNA biomarkers, but combining that with lipid biomarkers and then also culturing and in situ work as part of answering these questions. Now, one of the things that's intriguing about the haptophytes is that the marine taxa, at least, are incredibly conservative in their rate of change in the ribosomal RNA molecule, so much so that it was thought that there was only a single species present in the marine environment. That's changing with the advent of genomics, but it was really a real dilemma in terms of why these are so non-diverse, so to speak. Whereas in contrast, the freshwater taxa are amazingly more diverse, especially at the small subunit level, so much so that there have been recent discoveries of what were thought to be different and unique species of this group. But it turns out that part of it was just a disconnect between the sort of geology literature and the biology literature in terms of recognizing unique species that are present. But this group one is one that I mentioned earlier that we're especially interested in part because they seem to be very abundant in many different types of lakes. And this particular group is a very important group in terms of being able to apply some of the temperature proxies that we're interested in developing. Now, in terms of biomarker development, what we have found is that the small subunit ribosomal RNA gene actually breaks down when we try to resolve some of the relationships between these group 1 haptophytes that appear to be mostly found in fresh waters. Now, unfortunately, despite the fact that other members of the haptophytes that produce these alkanone molecules are cultivable, this particular group we have not been able to bring into culture yet, and probably because we haven't tried hard enough. But in the absence of that, we're trying to find markers, additional markers that we can use that will better resolve whether or not these are really a coherent group and monophyletic, or if in fact there are different lineages within this particular group of haptophytes. And one of the schemas that we're trying to use is one where we can go from the small subunit ITS and then to the large subunit to capture a larger diversity of these organisms that we hope will help resolve their phylogenetic placement. And ultimately, we want to be able to combine these gas chromatograph signatures of the different types of lipids that are formed, which we're finding out through novel methods and refined methods are becoming more and more useful in the geological record with the actual phylogeny of the organisms and ultimately genomics in resolving some of the relationships between them and also the relationship between how these two are linked. Okay. I'd like to segue now into the other group of protists that I'm working on, which include macroalgae, such as this one here, this brown alga, which is the namesake of the Sargasso Sea, which is located in the North Atlantic. And it was actually sargassum was first discovered by Columbus himself, who thought that it was an indication that the ships were near land because he encountered crabs that were associated with the weed. But if one looks very closely at sargassum, there's this whole really amazing invertebrate community that hangs out on the surface of the sargassum. And so at this point, you might be asking, well, what does sargassum have to do with protists? Well, it turns out that sargassum is itself a protist because it's more closely related to things like diatoms than any of the multicellular groups that we know, particularly the plants, which is sometimes thought to be an aquatic plant. But it really is phylogenetically distinct, and it is among the diversity of microbes that we understand. Now, and this is in a group called the Stramenophiles. Now, in terms of the question of sargassum and its diversity, it really is in and of itself an entire ecosystem. So these are 10 endemic taxa that call sargassum their home. Many of them are very cryptic and will hide within the sargassum itself. So it's more than just a macroalga, it's really an entire floating ecosystem. There's about 350 species of sargassum, and only 2 of them are responsible for being effectively the golden floating rainforest of the Sargasso Sea, in that they are asexual and also have only a floating form. The rest are benthic. Recently, however, the sargassum has gotten a bad rap, and that's because it is showing up in large quantities on various beaches. It is suffocating harbors and has really gone from being a great thing to too much of a good thing, so to speak, in terms of the impact it has made both economic and social and otherwise in predominantly Caribbean areas, but now most recently on the west coast of Africa and also Brazil. Now, my interest in sargassum originated from partly being the organizer of a curriculum that was designed at looking at conservation and biodiversity in the Atlantic waters through which sargassum is resident. And we were developing a suite of biomarkers that could be used with sargassum for the sake of student projects because we thought it would be a really nice project. But it turns out that sargassum is quite challenging to work with, both at the sort of DNA level, but also in terms of designing unique primer sets. But the question of being able to differentiate between the different species became especially important when colleagues at the Sea Education Association started publishing some of their archive data on sargassum and realizing that this inundating form that's accumulating in the Caribbean actually is a different morphotype. And it is one that was originally described by Parr in the 1930s to be incredibly rare. And so one of the things that is really kind of capturing our attention right now is whether this new genotype, it turns out, that's appearing in the Caribbean and in other parts of the South Atlantic is a natural range expansion or if it's a climate change related bloom. And there are various hypotheses out there that are targeted at understanding that question. The first reports in Brazil came in 2011, and this is the first time Brazil has ever actually had sargassum present, floating sargassum present in its waters, and it is unfortunately affected one of the few marine protected areas off of the northern coast of Brazil. So in order to attack this problem of whether we could genotype these different sargassum species, we started with CO1, which was unsuccessful at differentiating between the 2 different forms of what has been known to be called a natans 1 and natans 8. These are the 2 floating forms that are recognized, at least right now, genetically. We went to the herbarium at the Yale University to actually look at Parr's samples and confirm the morphology is correct in terms of differentiating between the different forms of sargassum. But then we ultimately needed to go from the CO1, basically targeting single marker genes, all the way to the entire mitochondrial genome. And what this tree shows here is a mitochondrial genomic tree of all available sargassum species that have sequenced mitochondrial genomes. And when we did this, we were able to differentiate the 2 natans. Natans 8 is actually the one that's responsible for these inundations. But again, it was really challenging, and it took sequencing the entire mitochondrial genome. And still, very, very few differences, but stable differences are present between the different genotypes that we looked at. So we recently published this work. We've also been looking at chloroplast genomics, and I partly included this just to acknowledge my co-authors on the study and thank them for their efforts in helping to resolve this issue. So we're moving forward with better trying to understand the source of the inundations and looking at different genotypes in the context of population genomics of the group. And I'm excited to say that as part of my new position, I'm going to be going out into a cruise that will sample this water that's in between Africa and South America called the North Equatorial Recirculation Region, which is hypothesized because of input from major river flows of the Amazon and the Congo to be help enriching these populations of sargassum that are causing these problems. So we're going to try to test that. So if I look into my crystal ball, what I'm seeing is that there really is a need to sequence more protist genomes. I think that marker genes have their place in differentiating between different types of protistan groups, but ultimately there are some groups that a single marker or even multiple markers are not going to provide the necessary resolution to differentiate between species. And this has certainly been my experience in delving into the two groups that I shared with you today. There was a recent paper that was published, and I believe in Nature, Ecology, and Evolution by Sibbald and Archibald, and this is sort of echoing this need to have more genomes sequenced within the protistan world, and very few have been sequenced thus far, and they're very biased in which ones, mostly parasites, or ones that cause human disease. But in order to really appreciate the role that protists play on so many different levels in the ocean and in any environment, I think we need to get a better handle on their genomic diversity as well as their genetic diversity. So I'm going to end there and leave you with my favorite quote from a famous scientist from the Marine Biological Laboratory, and thank my funders and my institutions that have supported this research over the past decades. Thank you for your attention.
Thank you, Linda. Are there any questions? We have like 2 minutes.
Thank you for this very nice introduction and presentation of Protists that we love, all of us.
Yes.
Of course. And there's only one question I had very specifically for your Haptophytes part.
Yes.
Did you think about using this for the fossil DNA kind of study, because you have lipids and you could have DNA? What do you think about this?
Yes. So in fact, some of our work that we've done in Greenland, where we first detected this this genotype, was done on effectively fossil DNA. So environmental DNA that was preserved. Now, many of the groups that are part of this group 1 that was under-recognized for so many years are, in fact, we think, naked. They don't form any kind of shell or hard part, so that they won't form traditional fossils, but they do form lipid fossils, and that is part of what we're doing. Yeah, that's a great question. Thank you.